This call is now being recorded. Okay, let me know. Can you see the screen?
Yes,
Just
ma'am.
clear. And my voice is also audible?
Yes, ma'am.
Yes, ma'am.
Okay,
Yes,
okay.
ma'am.
Yeah.
Yes, ma'am.
Thank you. Now, I will continue explaining, and at the end, you can ask me your doubts. If you have any doubts, you can note it down in the notebook, or even whenever I'll be telling you something new, which you find it new, terminologies, explanation, there in the notebook, either you write, or there for that particular word, you write it down in the textbook itself. Okay. Now, enzymes, next and the last part of this chapter, a last biomolecule which we are going to discuss right now, that is enzymes are simple or compound proteins acting as specific catalysts. Now, there are different definitions, but the meaning will be the similar, same. So they are defined as organic substances capable of catalyzing reaction in the living systems, or enzymes are biomolecules which contain proteins of high molecular mass and specific reactions. So enzymes are all proteins, but you remember that all proteins are not enzymes. Only those enzymes are considered where they are having the catalytic properties. Now you know the meaning of catalyst, which is you have learned already in other subject like chemistry, that is helpful for speeding or increasing the rate of reaction but doesn't take part directly in the reaction. So they are considered to be catalysts. So enzymes are considered to be biological catalysts which are involved in so many biological biochemical reactions. So catalysts are substances that increase the speed of a chemical reaction. It's not the permanently change. It's not permanently change, nor does it cause the reaction to occur. That is, a catalyst can increase the speed of the reaction, but cannot cause the reaction. Okay, so it is responsible for increasing the rate of reaction, increasing the speed of the reaction, and also it doesn't take part in the reaction. It is not responsible for that particular reaction. It cannot be the cause of the reaction if it would not occur in the absence of catalyst. Since catalysts are not used up, they can be used over and over again. So once the role is over, again they can be used. So enzymes can be used again and again as biocatalysts. So what is the biochemical nature of enzymes? These are all proteins except ribozymes. They are distinguished from other proteins by catalytic action. As I told just now, that these enzymes are the proteins. All enzymes are proteins, but all proteins cannot be the enzymes. Only those proteins with the catalytic property action only can be considered as enzymes. Yes, any doubt? Okay, someone's microphone is on, I think, so keep it off if anybody's microphone or a video is off. I am on the presentation mode, cannot see. So the
Sorry, ma'am.
catalytic power of enzyme is dependent upon primary, secondary, tertiary, and quaternary structure of the protein molecule. Change can affect enzyme activity. So catalytic power of enzyme is dependent on the structure and the nature of the molecule, that is protein, whether it is of primary model, secondary type, or tertiary or quaternary structure of protein molecule. Depending on that, the power of enzyme, that is the catalytic power of enzyme, is dependent. So change can affect enzyme activity. Any change in it then that can affect enzyme activity. So what are the enzymes? These are the organic catalysts produced by an organism. The reactant is an enzyme catalyzed reaction, is called substrate. Now there are two things you need to remember always. Okay, so this is what? This is the enzyme. This is the substrate, which are shown by different colors purposely. This is the substrate on which enzyme can act, but this is the area, or this is the place where this reaction can take place when this substrate comes in contact with this area of the enzyme. So this area is called as active site. Okay, so enzyme changes its shape slightly as substrate comes in contact with the enzyme. So though little change in the shape of the enzyme is observed, here you can see in the first diagram, but when the substrate comes in contact with the enzyme, it adjusts its shape in such a way that the substrate can fit in the active site of the enzyme. And this way, the product or the complex called as enzyme substrate complex is formed. And after that reaction is over, again these substrates can be removed. They can be worked as a, they can be considered as a products. Now we can see in this reaction in this diagram. So products leaving the active site of enzyme. Once the product is formed, this is also called as enzyme substrate complex. The entire structure is called as enzyme substrate complex. Here it is done. And after the reaction gets over, here the product is formed, then they get separated from this site of the enzyme. So what is active site? In the same diagram I showed you, explain you that the site of the enzyme or the area of the enzyme where the substrates comes in contact with the enzyme, that particular area is called as active site. So small portion of the molecule that is responsible for the catalytic action of the enzyme. This is what the perfect definition for active site you can consider. So the small portion of the molecule that is responsible for the catalytic action of the enzyme is called active site. So substrate, active site, and enzyme substrate complex that you need to remember. Now step by step we will discuss the chemical nature of enzyme. Simple protein enzymes. First category or first type of enzymes is these are all only simple proteins. So those enzymes which contain only simple proteins, they are the simple protein enzymes. For example, urease, amylase, papain, these are some of the enzymes which contain only proteins. But complex protein enzyme means what? These contain conjugated proteins. That is, they have a protein part called apoenzyme and a non-protein part that is called as prosthetic group associated with the protein unit. And the two parts constitute that is called as holoenzyme. Now, 3 points or 3 words you need to remember. First is what? Apoenzyme. Second word is what? Prosthetic group. And the third word, what it is called as holoenzyme. Okay. So apoenzyme means what? Apoenzyme is the protein part. Prosthetic group is non-protein part of the enzyme. And together is called as holoenzyme. So conjugated protein enzymes, they contain the protein part, which is called as apoenzyme, and non-protein part that is called as prosthetic group. And together what it is called? That is called as holoenzyme. So these two chemically, these two forms are observed. Now conjugated protein enzymes on hydrolysis, what they give? Protein part that is called as apoenzyme and non-protein part that is called as prosthetic group. So holoenzyme is nothing but the apoenzyme, and it prosthetic group may be coenzyme or cofactor. Now we will discuss what exactly that coenzyme means or cofactor means. So the activity of an enzyme depends on the prosthetic group that is tightly associated with the apoenzyme. But sometimes the prosthetic group is loosely bound to the protein unit and can be separated by dialysis and yet indispensable for the enzyme activity. In this case, this dialysable prosthetic group is called as coenzyme or cofactor. Now, just now I was mentioning what this prosthetic group is. Prosthetic group may be coenzyme. That means it is the organic group which is tightly bound to the protein part. But this prosthetic group can be the inorganic molecule, inorganic in nature, bound to the protein part, that means apoenzyme part, but which is loosely bound. So that is called as cofactor. So what is the difference between coenzyme and cofactor? This part, which is protein part, is common, that is called as a coenzyme, but prosthetic group may be coenzyme or cofactor. Now what is coenzyme and what is cofactor? Coenzyme is the organic part and cofactor is the inorganic part. So you remember, organic part is coenzyme, which is tightly bound to the protein part. But cofactors are generally metals and these are inorganic in nature and they are loosely bound to the protein part, that is apoenzyme part. So these 2 points, the differences between coenzyme and cofactor, you need to remember, which are considered to be prosthetic group. So here again, it is mentioned that is non-protein organic, that is low molecular weight and dialysable substance associated with the enzyme function is known as coenzyme. And they are often regarded as the second substrate or co-substrate since they have affinity with the enzyme comparable with that of the substrate. Means they have the affinity with the enzyme as compared to the substrate. Now, just now we discussed about the substrate enzymes complex, right? Same way, that part of the enzyme is very important, that is considered to be coenzyme, which is tightly bound, though it is non-protein part and it's playing the role in the substrate complex form. Okay, enzyme substrate complex. So the types of coenzymes, for example, B complex vitamin and coenzymes and non B complex vitamin coenzymes. These are the enzymes, coenzymes example. These are the two types. One category is B complex vitamin coenzymes and the another is non B complex vitamin coenzymes. These are the two types. Now, cofactors. These are non-protein, inorganic, low molecular weight and dialysable substances associated with the enzyme function, and they are generally metal ions. And these metal activated enzymes, these are the enzymes with the metals form a loose and easily dissociable complex. Now, as I told you that these are loosely bound to the protein part, that means apoenzyme, but coenzymes they are tightly bound. Both are non-protein, but coenzymes are organic, low molecular weight, and cofactors are inorganic. Coenzymes are tightly bound, these cofactors are loosely bound. Then, and generally these are all metal ions. Now these some of the examples are ATPase, where Mg plus 2, Ca plus 2, enolase, Mg plus 2. These are the ATPase, where in ATPase is the enzyme generally. Now how this nomenclature comes, that we will discuss right now. So ATP is synthesized, or that enzyme which is responsible for acting on the ATP, that is adenosine triphosphate, that is called ATPase, and where these are the cofactors, where Mg plus 2 ions and Ca plus 2 ions, these are the metals, so these are the cofactors. Metalloenzymes is one more category, where in this metal ion is bound tightly to the enzyme and not dissociated. Now this is the difference between the cofactors and metalloenzymes. Cofactors, they contain inorganic substance and most of the times these are metals, but these are loosely bound, they can be dissociated. But metalloenzymes, they also contain metal ions, but they are tightly bound to the enzyme and they are not dissociated, they cannot be separated, they are not dissociated from the enzyme. So this is the difference between the cofactors and metalloenzymes. Cofactors also contains the metals which are light, very loosely bound and can be dissociated, but metalloenzymes are also metal ions, but they are tightly bound to the enzymes and they are not dissociated. Now, for example, alcohol dehydrogenase, carbonic anhydrase, alkaline phosphatase, these are some of the enzymes. Phenol oxidase, where copper is bound, then pyruvate oxidase, manganese is bound, xanthine oxidase, molybdenum, cytochrome oxidase, iron and copper are bound. The iron and copper, these which are mentioned in the bracket, these are the metal ions. These are considered to be metalloenzymes. These metals are bound to the enzymes tightly, which cannot be dissociated, which cannot be separated. Now, how the enzymes are named and accordingly classified. Nomenclature is what? Giving name. Every living as well as non-living organism or the object or the substance is known by some name. So giving the name is what? Nomenclature. Nomenclature is the way of giving the name to that particular living organism or non-living thing. So how these are named? Substrate acted upon the enzyme. This is the first category or the way of giving name to the enzyme. First means on which the enzyme is acted, that particular substrate's name comes, and then ASE is added at the end. So in this way the names are given to the enzyme. So this is the first way. For example, name the enzymes by adding ASE in the name of the substrate catalyzed. Carbohydrates means what? Here carbohydrate is a substance on which the enzyme acts. So what is the name? Carbohydrases. Proteins, substrate on which the enzymes are acting, that means proteases. Lipids on which the enzymes are acting, that these are lipases. Nucleic acids is the name, is the substrate when it is known as nucleases, like that. A few of the names were given even more specific, like maltase acting upon maltose, sucrase acting upon sucrose, urease which act on urea, like that. These are very specific. The name of the substance is mentioned, and at the end on which that a particular substrate it acts, the ASE is added at the end. The second category of giving the name is what? Type of reaction they catalyze. Means the enzymes, these enzymes are named by adding ASE in the name of the reaction. For example, hydrolysis means the enzymes which perform or which catalyze hydrolysis. They are called as hydrolases. Isomerase means isomerization reaction is performed. Oxidases which catalyze oxidation. Dehydrogenases catalyzing dehydrogenation. Transaminase which catalyze the enzymes which catalyze transamination. Phosphorylates catalyzing the reaction phosphorylation. So in this way, the type of reaction which they catalyze, and accordingly the names are given to the enzymes. So third category is what? Where substrate on which the enzyme is acted upon, and also the type of reaction which the enzymes catalyze. So both the points are considered, the first and the second one, and accordingly also the names are given. The names of the some enzymes give clue of both the substrate utilized and the type of reaction they catalyze. For example, succinic dehydrogenase means what? It catalyzes dehydrogenation reaction of the substrate succinic acid. Substrate is succinic acid, and the reaction which they catalyze is dehydrogenation. So accordingly the name of the enzyme is what? Succinic dehydrogenase. Then L or alpha glutamic dehydrogenase, which catalyzes dehydrogenation reaction involving L-glutamic acid. So accordingly the names are given, and this is in short, in brief, you can remember the classes of the enzymes and reactions they catalyze. That means oxidoreductase, which catalyze oxidation reduction. Transferases, transfer group of atoms. Hydrolysis, hydrolysis, that is cleave or add, addition of water. Lysis, which cleave without adding water. Isomerases, that is rearrangement of the atoms. Ligases, which combine the molecules using ATP. So don't get confused between lysases and ligases. Lysases, these are without adding water, they cleave the reaction. Whereas ligases, they join, combine. Ligase, they help in combining the molecules with the help of ATP. So these are some of the classes with the of the enzymes. So there are two more categories you need to remember, that is endoenzymes and exoenzymes. The name itself suggests, means what? Endo is what? Inside, within the cell, and exo is what? Outside the cell. Now enzymes that act within the cells in which they are produced, they are considered to be endoenzymes or intracellular enzymes. For example, plant enzymes, metabolic enzymes. These are all the type of enzymes where they produce these enzymes, plants produce the enzymes, and within the cells they are acting. So these are intracellular enzymes, or they are also called as endoenzymes. And then enzymes which are liberated by living cells and catalyze useful reactions outside the cells in its environment, then they are considered to be extracellular enzymes or exoenzymes. So these are some of the So bacterial enzymes, fungal enzymes, digestive tract enzymes, these are some of the examples where these bacteria release enzymes, fungi release these enzymes, then our digestive tract, maybe human beings or whatever the higher organism, the digestive tract which release enzymes. So their action is outside the cell where they are produced. Okay, so such type of enzymes are considered to be extracellular enzymes or exoenzymes. Okay, then what are the characteristic features, these few important points about enzymes? Enzymes, they are called as biological catalysts. Always remember, they are present in all living cells. Then the next important thing is that they are made up of proteins. Then enzymes convert substrates into products. Just now we discussed the substrate enzyme complex, and then whatever the products are produced, they are out. Then the next is what? Increase the rate of chemical reaction. What is the biological catalytic property of the enzyme? So why they are called as catalyst? Why biological catalyst? Because in all type of biological reactions, they help, they play the role of catalyst which help in increasing the rate or the speed of the reaction. So they are called as biological catalyst. And then they remain unchanged by chemical reaction. Though, so they are not directly taking part or involved in the reaction. They are just responsible for speeding up or increasing the speed of the reaction. So they remain unchanged at the end of the reaction. So can be used again and again. So we'll discuss these properties one by one. High degree of specificity for their substrate. All the enzymes, they are having high degree of specificity for their substrate. So each enzyme will catalyze only one particular reaction. Now in this case, in this diagram, you can see this is the enzyme and this is the substrate. So the, as per the active site of the enzyme available, that enzyme can fit only that part of the substrate which can fit that active site. And so these are very specific. That enzyme, one enzyme cannot react with any of the substrate. It is very very specific to the substrate. It can react with only a specific type of substrate. You can see in the diagram, it has not reacted or it is not taking part or it is not showing any reaction with this part of the substrate, but it can fit only in this specificity, you can say. Then enzymes are not used up in the reaction they catalyze. They can be used again and again. Now in this reaction, we have where you can see this is the enzyme and this is the substrate, where this is the enzyme substrate reaction, and after this is done, then what happens? It is released. This enzyme is released. This enzyme is released to use again for the next substrate to act upon. So in this way, it can be used again and again. Here you can see in this diagram at three places. Here this is the enzyme where it is acted with this, acting with the substrate at one particular place, forming the enzyme substrate reaction complex. And once the product is formed, it is released out, and then again this enzyme is ready to react with the another substrate. Okay. um, See, I'm with the full network, so I don't know why my voice is cracking. I will try, but okay. Now the next is when enzymes react, they combine with substrate to form ES complexes. Already I showed you in the previous diagrams. So this is what, suppose the substrate and this is the enzyme and this is the active site of the enzyme. So you can see this is what enzyme substrate complex. In this way it reacts. Now you can see one more thing, only small amount of enzyme is needed to catalyze a lot of substrates. In the previous diagram also you can see here is the small area that is considered to be the active site, where it reacts with the substrate. So large area and number of enzymes as well a large area remaining area of the enzyme is free to react with the different substrates. So small amount of enzyme is needed to catalyze lot of substrates. This is what you need to remember. Then enzyme concentration is also very important. Enzymes are fast acting. They have a high turnover number. They can convert many molecules of substrate per unit time. Means within a small time, within a short time, n number of products can be produced when the enzymes come in contact with the substrates. So its rate is very fast. It acts very fast. So that's why they have a very high turnover in case of substrate complex formation. Then enzymes are affected by changes in temperature and pH. This we will discuss in detail in the next part also. But you remember, with the help of this diagram, you can see when the temperature is increased, the rate of the reaction also increases. But up to a certain degree, that means up to at the optimal temperature, its rate is highest. When again it goes down, the rate goes down. Okay. Same way, in case of pH also, the rate of the reaction, that means enzyme activity is maximum at optimum pH. Enzyme activity is maximum at optimum pH as well as optimum temperature. So this you need to remember. Now, many enzymes work only if a chemical called cofactor is present. Now, we have already discussed this part that if cofactor is, how it is playing the role, it is loosely attached. It might be the metal or non-metal part, but it is inorganic substance when it is loosely attached to the protein. But without this cofactor, sometimes enzymes cannot work. So many enzymes work only if the chemical called cofactor is present. Okay, so this is the binding site of the cofactors. L1 and L2, they are shown to be cofactors, and this is the protein part, and this is the inorganic molecular part, low molecular weight compound, which is in non-protein part. Enzymes catalyzed reactions are slowed down or stopped by inhibitors. This part again we will discuss in detail. That means what? There are some substances which inhibit the reaction of the enzyme they catalyze. When these substances come in contact with the enzymes, they cannot show their catalytic property. So these are considered to be inhibitors. So such type of inhibitors are responsible for slowing down the reaction they catalyze, or they can stop. These inhibitors are responsible for not performing the reaction. That means they can stop the reaction also. Now, how the mode of enzyme action is there, we will see. There are two things. The mechanism of so-called as Fisher's template theory, and the second is what? Induced fit mechanism or induced fit theory, or this is also known as Koshland's model. Now, what are these? One by one, we will discuss. So, when lock and key model, which was first of all proposed by Emil Fischer, very first model to explain an enzyme catalyzed reaction. According to this model, the structure or conformation of the enzyme is rigid. That means the substrate fits to the binding site just as a key fits into the proper lock or a hand into a proper glove. Thus, the active site of an enzyme is rigid and pre-shaped template where only a specific substrate can bind. Now, let me explain you with the help of this example, this diagram. Now, you can see here what does this model explains that if the this active site of the enzyme, according to this model, that is lock and key model, why the name is given as lock and key. Any key cannot open any lock, isn't it? For a lock, a key is also fixed. That key can open only a particular specific lock only. That means how a key fixed or it fits in that particular lock only. In the same way, the substrate which can be considered as a key, which can fit only that active part of the enzyme which can fit the substrate. According to this theory, this active site is rigid. That means it cannot change its shape. It is fixed, it is rigid. That substrate only can fit in the active site of the enzyme, uh, which can accommodate or fits in the size. So this site, that means active site of the enzyme, is rigid and cannot change as per this model. That means as per this theory, which is called as lock and key theory. And accordingly, that substrate only will fit in the enzyme, active site of the enzyme, which is having that particular shape and which can only fit as per the shape and forming the enzyme substrate complex. But this model was not accepted. Why? Because it does not give any scope for the flexible nature of the enzyme. The which is enzymes, fails to explain many facts of the enzymatic reactions, and it does not explain the effect of allosteric modular. Now you just remember, this is not accepted because it cannot prove the rigid nature of the active site of the enzyme. So that's why this theory was not accepted later. But it has the importance because this was the first theory to explain the role of or the reaction of the substrate with the enzyme. So it is also having important role. Now the second is what? Induced fit theory, or this is also called as Koshland's model, or this is also called as induced fit mechanism. Koshland proposed this model. This is the name of the scientist, just like that previous name, Fisher, Emil Fisher. So here is the Koshland who proposed this model. The active site is not rigid and pre-shaped. According to this model, that means according to induced fit mechanism or theory, the active site of the enzyme is not rigid or it is not pre-shaped. But when the substrate comes in contact with the enzyme, that active site of the enzyme, that active site or the enzyme induces or it adjusts its active site in such a way that the substrate can fit in that active site. So it induces a fit or a conformation change in the enzyme, resulting in the formation of a strong substrate binding site. Furthermore, the appropriate amino acids of the enzymes are repositioned to form the active site and bring about the catalysis. Now you will not understand the last point, but let me tell you that amino acids are what they are made up of. Proteins are made up of amino acids, and what the enzymes are, they are made up of proteins. So that's why you just remember, as the appropriate amino acid number, it comes in contact with it, so accordingly it changes its shape in its structure. Protein models, if you remember, primary, secondary, tertiary, accordingly, at first only we discussed that as per the structure of the protein, the enzyme action is dependent upon that particular structure of the protein. So in this way, so you remember this thing, that this is considered to be most accepted one theory, as it shows and proves that to some extent it is that active site of the enzyme is not rigid or pre-shaped, but when the substrate comes in contact with the active site of the enzyme, it adjusts itself in such a way or fits the substrate in that particular active site and forms the strong substrate binding site. This model was accepted because it has sufficient experimental evidences from the X-ray diffraction module studies. Okay, and it explains the action of allosteric modulators and competitive inhibitors on enzyme. Competitive inhibition, I will not explain you right now the allosteric nature or modulators, but we will be having discussion about the competitive inhibitors. We just spoke about the inhibition or inhibitors. We will discuss that part. So here you can see, as per this model or the theory, that induced fit mechanism. See, this is the active site of the enzyme and this is the substrate. When this substrate comes in contact with the active site of the enzyme, this enzyme changes its shape. That means active site changes its shape. It is not rigid. So it is not formed or preformed structure. So what it is done, that active site changes its shape and it fits with the substrate in such a way that it forms a strong binding site with the substrate. So this is what the most accepted one theory. Now, as we were talking about the factors affecting enzyme action, again we will discuss few more points. We have already discussed in the properties, but still we will separately discussing these factors affecting enzyme action. What are the factors? First is what? Concentration of the enzyme. In the With the help of this diagram, the plot, you can understand as the enzyme concentration increases, its rate enzymes activity also gets increased. So as the concentration of the enzyme is increased, the velocity of the reaction proportionately increases. So the property of enzyme is made use in determining the serum enzymes for the diagnosis of the diseases. So this is helpful. Now how it is helpful, that we will discuss later. So you just remember, as the concentration of the enzyme increases, its speed or the rate of activity of the enzyme, velocity of the enzyme also gets increased. Second is concentration of the substrates. As the substrate concentration increases, automatically the enzyme reaction also is increased. So increase in the substrate concentration gradually increases the velocity of the enzyme reaction within the limited range of substrate levels. Now, how this is done, hyperbola or distinct phases, you have seen in the uh, previous diagram, but this is what the this is considered to be the hyperbola. Now here the substrate is increased. As the substrate is increased, its velocity also gets increased. It's increasing up to a certain limit. It is increasing with a high speed, but after some particular period, though we are increasing the substrate molecules, its rate slows down, and at a particular point it remains same. It doesn't increases the rate of the enzyme action. So this is what called as hyperbola, where it shows with the help of this reaction, this diagram, that though there is increase in the concentration of the substrate, enzyme activity is increased up to a certain level. First of all, it increases its speed with a particular rate, but at after after a particular point, the enzyme activity doesn't increase with that particular speed, but at a certain point, though you are increasing the amount of the substrate, the concentration of the substrate though is increased, it doesn't increases the rate, its enzyme activities that doesn't increase. So up to a certain level only you can consider. So in the previous explanation, what is given is, when the velocity of the reaction is almost proportional to the substrate concentration, the rate of the reaction is said to be first order with respect to substrate. But when substrate concentration is much greater than concentration of the enzyme, the rate of the reaction is independent of the substrate concentration and the reaction is said to be zero order. You got this point, which you will not be understanding with this explanation, but what I showed you with this diagram. At a certain limit, it increases. That means as the substrate concentration is increased, the velocity means enzyme rate is also increased. But after a certain point, the rate slows down and then it stops. Though you increase the substrate concentration, it doesn't affect the velocity of the enzyme activity. So this is what shows this diagram, this plot. The next is what we were talking already about this, that is the effect of temperature. With the help of this diagram, it's very clear. As the temperature increases, the rate of enzyme also increases, but up to a certain limit. That means at optimal temperature, the rate of enzyme activity is maximum. And this is the bell-shaped curve, which is shown that this is what the optimum temperature. At the optimum temperature, the enzyme activity is highest. So temperature coefficient is defined as increase in enzyme velocity when temperature is increased by 10 degrees Celsius. For majority of enzymes, this is between 0 degree and 40 degrees Celsius. But optimum temperature is considered to be 40 to 45 degrees Celsius. But some of the enzymes, they work below 40 and above 45. So there are some of the enzymes, few enzymes like venom phosphokinase, muscle adenylate and inates. These are active even at 100 degrees Celsius. Even some plant enzymes like urease have optimum activity around 60 degrees Celsius. So there are some exceptions, but in general, the activity, that is the temperature which is considered to be optimum, is 40 to 45 degrees Celsius, and this is what the temperature, optimum temperature enzyme activity is maximum at the highest speed. The enzymes are exposed to a temperature above 50 degrees Celsius, denaturation leading to dearrangement in the native tertiary structure of the protein, and active sites are seen. And majority of the enzymes become inactive at higher temperature. So as the temperature increases, denaturation of the enzymes takes place. That means its original forms gets changed. As you know that enzymes are made up of proteins, so there's the proteins are made up of amino acids. As the rate of the this temperature increases, automatically their original form, shape, whatever tertiary model, it changes to secondary or primary, like that they are changed. And generally above 70 degree they become inactive. Same here also, when pH is optimum, at the optimum pH, the maximum activity of the enzyme is observed. Below and above this pH, the enzyme activity is much lower, and at extreme pH, the enzyme becomes totally inactive. So this is what the specificity for temperature, specificity of the enzyme with the pH, specificity of the enzyme with the substrate, and even enzyme concentration. These 4 points we have discussed. And what is that? The range is given here. That is, most of the enzymes of higher organisms show optimum activity around neutral pH, and neutral pH is 6 to 8. But there are, however, many exceptions like pepsine, which act in between 1 to 2, acid phosphate between 4 to 5, and alkaline phosphatase in between 10 to 11. These are the pH mentioned in the bracket. Enzymes from fungi and plants are most active in acidic pH, that is between 4 to 6. Hydrogen ions influence the enzyme activity by altering the ionic charges on the amino acids, particularly at the active site and the substrate. So these are some of the exceptions given where they work below pH. Neutral pH is considered to be between 6 to 8, and most of the higher organisms, they are active at this neutral pH. But pepsine, then phosphatase, alkaline phosphatase, these are some of the exceptions. And enzymes from fungi and plants generally are active at acidic pH 4 to 6. Remember these points. Now, what does it mean, enzyme inhibition? That we will see. Enzyme inhibition means what? These are the inhibitors which are defined as the substances which bind with the enzyme and brings about a decrease in the catalytic activity of that enzyme. Inhibitor may be organic or inorganic in nature, and there are three broad categories of enzyme inhibitors. Now we will discuss these one by one. Reversible inhibitors and irreversible inhibitors. Inhibitors what? Which inhibits the reaction. Now, what is the role of enzymes? What is the property of enzyme? They catalyze the reactions. But when the enzymes come in contact with such substances, then the rate of the reaction is slowed up or it can be stopped. There is a slowing down of the rate of or the speed of the enzyme activity. They catalyze, or these activity or the reactions even cannot take place. They are stopped. What is done? What is mean by, or does it mean by reversible inhibition? Reversible, the name itself suggests. Okay, the inhibitors bind non-covalently with enzymes. Enzyme inhibition can be reversed if the inhibitor is removed. This is so simple, you remember. Reversible means if when inhibitor comes in contact with the enzyme, it affects the rate of the enzyme. But when inhibitor is removed, automatically its activity starts. Again, enzyme acts as usual. So this is what reversible inhibition. But again, it is divided into two categories, two types. That is competitive, and the second is non-competitive. Now, what is that competitive inhibition? With the help of the diagram also you can understand. Here, this is the enzyme, this is the active site, this is the substrate, and this is the inhibitor. Okay, you can find the shape of the substrate and the shape of the inhibitor and the shape of the active site of the enzyme. Okay. Now, if you see, both the substrate and inhibitor are little bit similar in shape, which can fit the active site of the enzyme. So in this way, their shape is competing each other. Substrate E and inhibitors both are in competition to fit in the active site of the enzyme. So these are called as competitive inhibitions, where the shape of the inhibitor and the substrate is similar. If substrate comes in contact, then substrate enzyme complex will form. If inhibitor comes first in contact with the enzyme, this active site, then this inhibitor complex will be formed. That is what enzyme inhibitor complex will be formed. So the rate of the inhibitor inhibition depends on then concentration of the substrate and inhibitor. If the concentration of the substrate is more, substrate will bind to the active site, and then enzyme substrate reaction will be formed, the product will be produced. But if the concentration of inhibitor is increased, it is more, then rather than substrate, inhibitor will first fit into the active site of the enzyme, and then it will inhibit the activity of the enzyme itself. So the inhibition can be reversed by increasing the concentration of the substrate, that you can remember. So if you increase the concentration of the substrate, the inhibitor cannot come in contact with the substrate, and in this way you can uh, see that this is the competition because of the similar shape of the substrate and the inhibitor, and that enzyme activity is inhibited. That means rate of the inhibition depends on the concentration of the substrate and inhibitor, as well as affinity of the inhibitor towards the enzyme. If that inhibitor is having more affinity than the substrate, then it will form the complex with the enzyme. So inhibition can be reversed by increasing the concentration of the substrate. If you increase the concentration of the substrate, automatically the inhibition rate will be reduced. Now, what is non-competitive? Now you can see in this case, the rate of the inhibitor depends on the concentration of the inhibitor. Mean it doesn't depend on the shape. Generally, competitive inhibition is for because of the similar shape of the inhibitor and the substrate. But here, the competition is not because of the shape, but rather than that, the concentration of the enzyme, inhibitor or the substrate. So the rate of the inhibition depends on the concentration of the inhibitor. If the concentration of the inhibitor is more, it will affect the activity of the enzyme. So that is very important. So this is what called as non-competitive type of reaction. Of the reversible type we are talking, one is here enzyme substrate complex is formed, the product is produced, enzyme is separated. When it comes in contact with the inhibitor, then here is this again, if inhibitor comes in contact with the enzyme, this is what enzyme inhibition complex. Then again, when substrate comes in contact, then the combined form is formed. Here also you can see in this reaction C, this is what this ESI, that means enzyme substrate and inhibition. If you can remove the inhibitor, then the rate will be increased, rate of the enzyme will be increased. So that's why it is said that it depends on the concentration of the inhibitor. If the substrate concentration increased, this inhibitor concentration will be reduced. So this you need to remember. These are some of the examples which are mentioned here. That is, various heavy metals like Ag, Hg, and Pb, they inhibit the activity of a variety of enzymes. These are some of the examples, metal ions, which come in contact with the active site of the enzyme, then they reduce the rate of the enzyme activity. The UES is, for example, highly sensitive to any of these ions in traces. If then these ions come in contact with the enzyme urease, then its activity is reduced or stopped. It affects the rate of enzyme activity. So heavy metals form mercaptides with sulfhydryl, that is, these are the sulfhydryl bond groups of the enzyme, and in this way the rate of the enzyme activity is affected. Now these are one more examples like the cyanide and hydrogen sulfide. They are strongly inhibitors, considered to be strong inhibitors, and they act on this the enzymes of iron containing enzymes like catalases and peroxidases. So these cyanide and hydrogen sulfide, they are the examples of inhibitors. What is the second category? That is irreversible inhibition. They bind covalently with the enzymes and inactivate them irreversibly. Means they cannot be again come to their original shape or position or the function. So these inhibitors usually are toxic, poisonous substances. Irreversible, these are the irreversible inhibitors which combine with or destroy a functional group on the enzyme that is essential for its activity. You remember, these irreversible inhibitors, they affect the enzymes which where they destroy the functional group of the enzyme with which it shows the activity. So these are, most of them are the poisonous substances. They are toxic. For example, lodoacetate, which is irreversible inhibitor of papain, and glyceraldehyde 3-phosphate dehydrogenase. So these are some of the examples which of this irreversible type of uh, enzyme inhibitors. Now this is DFP. This is also called as diisopropyl fluorophosphate. It's a nerve gas developed by the Germans during Second World War and irreversibly binds with the enzyme containing serine at the active site. And so these are very dangerous. So poisonous, toxic, and dangerous to the organisms and to the environment also. Few more examples are mentioned. That is organophosphorus insecticides. So these insecticides also, they are some of the examples where this toxicity to that particular species, toxic to particular animals. So they block the activity of this acetylcholinesterase. These are just examples mentioned. Then penicillin antibiotics, they also act as irreversible inhibitors of serine. So these are some of the examples of uh, what you can say enzyme inhibitors of irreversible type. So this is what about the chapter. And if you have if you want to have any discussion, you can ask me. If you want to ask any questions, whatever we have discussed, we have